It's my honor to introduce the next speaker this afternoon, Marsha Lester from the University of Pennsylvania. Marsha was an undergraduate at Douglas College at Rutgers and earned her PhD in chemistry as a student of George Flynn at Columbia University. I met her there originally. She's now the Edmund J. Kahn Distinguished Professor in the Department of Chemistry at the University of Pennsylvania. She's a member of the National Academy of Sciences. She's the chief editor of the Journal of Chemical Physics, and she's a fellow of the Physical Society and the Chemical Society, has won numerous prizes. And her research is a rigorous study of chemical reactions, quite rigorous, with novel spectroscopy. And she studied a number of important intermediates in the environmental processes. And this afternoon she'll be speaking to us about exploring uncharted regions of atmospheric chemistry reaction pathways. Marsha.
Well, so as everybody has already commented, it's not easy to follow that morning session. I want to start by with a bit of gratitude to the Dreyfus program. I was a teacher scholar a long time ago, but I also want to acknowledge the postdoctoral program in environmental chemistry. I've had the opportunity to have this award twice. To Craig Murray initially held it and is now an assistant professor at UC Irvine, and Joe Beams is now a tenure track faculty member at Cardiff University. And the award in 2012 was really important because that was actually the first funding of the project I'm going to tell you about today. So let's get started. I've had a fascination with the hydroxyl radical throughout most of my career. It's the most important oxidant in the lower atmosphere. And it initiates the breakdown of most hydrocarbons, including methane, one of the most important greenhouse gases. And it also, and most trace species that we emit into the atmosphere are broken down through their reaction with hydroxyl radicals. So I've studied it from the reactivity and the reactions that result from hydroxyl radicals. And the story I'm going to tell you today is about the formation of hydroxyl radicals in the atmosphere, because they constantly are being formed and removed, and they're present in the atmosphere at a very low concentration, only in the order of 10 to the 6 molecules per cc. It's very, very low in steady state. So we really care about how they're formed. So many of you may have learned in introductory chemistry classes that the chemistry is all about the solar photolysis of ozone that makes O-singlet-D, electronically excited oxygen atoms, that can then react with water molecules to make 2 hydroxyl radicals. And in the classical atmospheric chemistry textbooks, that is the mechanism that you would read. But it turns out that field studies, for example, this TORCH study that was done in London area in the summer of 2003, have shown that that's actually only accounts for about 1/3 of the hydroxyl radicals in the atmosphere, and that there are other processes that are important. One is the solar photolysis of nitrous acid, and that comes about often in high NOx polluted environments. But again requires sunlight. So both of the 2/3 that are on the left-hand side are solar-driven chemistry. And there's another third, and that's this on the right-hand corner here, and that's the reactions of alkenes, organic molecules with carbon-carbon double bonds that react with ozone to make hydroxyl radicals, and this does not require sunlight. Okay, so it's a non-photolytic process. Some studies have even indicated that as much as 50% of the hydroxyl radicals in the atmosphere in the summertime, when there's, as you'll hear, large biogenic alkene emissions and a lot of ozone, could be driven by this alkene ozonolysis reaction. So what I'm going to tell you today is that it sounds straightforward. But I'm going to tell you there's actually there's a lot of questions about this chemistry. In fact, and that will be my story today of how that chemistry actually works from a physical chemist perspective, okay? At nighttime, this reaction also occurs. There's less hydroxyl radicals in the atmosphere, but this is the chemistry that's also driving oxidation in the atmosphere. Okay, so many of you may be saying, why alkenes, and why could they possibly be so important in the atmosphere? Well, it turns out that after methane, they're the most abundant organic species in the atmosphere. Again, alkenes are carbon-carbon double bonds. And we all know that they're there. We're used to the smell from pine trees. That's pinene, alpha-pinene. And that's part of the class of terpenes. Isoprenes make up about 1/3 of the non-methane emissions. And then they're coming primarily from plant emissions, plants and trees. They also come about from biomass burning and fossil fuel emissions, and they tend to make smaller alkenes like ethene or propene or butadiene. And so those are the systems we're going to be interested in studying. Okay. So let me tell you about the basic chemistry. Again, I know there's a lot of organic chemists here, so it's a little bit simplified version of this, of how it happens in the gas phase. So we're going to take your just typical alkene, and it reacts with ozone. And the way it reacts is that ozone adds across the carbon-carbon double bond, and it makes what atmospheric chemists call a primary ozonide. And then that primary ozonide splits apart in 2 parts. It breaks an OO bond and carbon-carbon bond simultaneously, and it releases 2 fragments, a stable carbonyl compound, an aldehyde or a ketone, and also this carbonyl oxide species, this unusual zwitterionic structure that I have illustrated here that are called Criegee intermediates. And I'll tell you a bit more about them in a moment. They're initially formed highly energized, and Rudolf Criegee did his work in the 1950s and proposed this reaction intermediate. And the bottom line is it wasn't observed for the first time until 2012, and I'm going to tell you about how we're observing it and why it's so important. And how it converts through what I will show you to be an intramolecular hydrogen transfer reaction that ends up releasing hydroxyl radicals. And moreover, the story is going to be that this process, even under thermal conditions in the atmosphere, happens by quantum mechanical tunneling. Okay? So hold those thoughts. So the things that I want to tell you about are how does this reaction occur? We're going to look at it. It depends strongly on the substituents, R1 and R2. Even simple changes in substituents from a hydrogen atom to a methyl or a larger system can change the chemistry dramatically and particularly affects the yield. And all of the release of hydroxyl radicals is going to be in competition with bimolecular reactions that are taking place in the atmosphere, water vapor, SO2, NO2, that are going on simultaneously. So, previous studies, these kinds of systems have been studied for years and years in the atmosphere. In fact, they're even tabulated in IUPAC. What the yield is of OH from various alkene ozonolysis reactions. So if you start with ethene, the simplest alkene, and have it undergo ozonolysis, the OH yield, the OH radical yield, is very low, but it increases dramatically as you go to 2-butenes, or if you go to what we commonly call tetramethylethylene, where the yield of OH from ozonolysis can be really quite large. So I'll tell you more about the story of how we do these experiments later, but we have been able to directly start from this reactive intermediate, these Criegee intermediates, and show that we get a very similar increase in yield of OH when we go to substituted intermediates with methyl groups or larger. Okay, so this is the atmospheric chemistry view of this kind of chemistry. We have alkene ozonolysis. We make what is called the Criegee intermediate. it can undergo, it's formed initially energized. There's about 50 kcal per mole of excess energy, and it's released, some of it's released promptly, and so there's OH formed immediately. And then in the atmosphere, it's at atmospheric pressure, of course, there's collisional stabilization. And atmospheric chemists have hypothesized that there would be a stabilized Criegee intermediate present in the atmosphere as well. And then that stabilized intermediate could go on in these bimolecular reactions, and it would act as an oxidant in and of itself. For example, oxidizing SO2 to SO3 and then eventually sulfuric acid, reacting with NO2 in a similar way, and water vapor. I'll tell you more about that a little bit later. But very important in all of this is that these reactions are thought to be in the initiators for the nucleation steps, the initiators for formation of secondary organic aerosols. And aerosols are, of course, worrisome from a human health perspective, but they're also very important in climate change. And so here is chemistry starting from this reactive intermediate that is impacting in the atmosphere. Okay, and then on the other side, this now stabilized intermediate can undergo a thermal decay process and again release hydroxyl radicals. So what I'll be telling you about today are, well, first, how do you isolate or make or stabilize these intermediates if they're so important? How can you sensitively detect them? And then I'm really going to focus on how they rearrange to release hydroxyl radicals, which are so important in the atmosphere. What we'll do is we'll look at it in an energy-dependent way, giving the system specific amounts of energy and measuring the rate at which hydroxyl radicals are released. And we'll also extrapolate to under thermal conditions. And as I said already once, but I'll repeat again, this process involves quantum mechanical tunneling of the hydrogen atom in the hydrogen atom transfer process. Okay, so why has it been that this class of reactive intermediates has been hypothesized to be so important but never observed? And this is illustrated here for the simplest system, ethene plus ozone. It reacts actually rather slowly. It makes various types of intermediates. The Criegee intermediate, this Vitter ion that I'm going to be telling you about, could also make dioxarane, a ring-like system. It could also have a dioxymethylene type structure. And all of those are isomers. They all have the same mass, different configurations. And then they're consumed rapidly, either by rearrangement, for example, as I have illustrated here, to make formic acid, or alternatively, and formic acid has, again, an isomer, the same mass, but now a stable product. It falls apart to make other species, and it can also be consumed by bimolecular reactions with the reactants themselves, with the products, with other atmospheric species, or if you do this in solution, of course, with the solvent. So all of those places. And the bottom line is they had never been observed. Okay, so what happened? In 2012, there was a beautiful, a new way of generating these reactive intermediates. And it's totally different chemistry, but it makes the desired intermediate that it gives us a starting point for investigating the chemistry. And it's sort of simple, but it was unexpected, I think. If you start with diiodoalkanes and you photolyze them, we do this in the UV at 248 nanometers, it releases, it breaks a carbon-iodine bond, and it makes an iodoalkyl radical. And then that radical reacts with O2 molecular oxygen, and the oxygen replaces the iodine and makes the Criegee intermediate. Well, how do you know? There's all these different isomeric forms. They all have the same mass. How do you know you have it? And so this chemistry was first done by Craig Tages and co-workers at Sandia National Laboratory. And they used the advanced light source at Berkeley to have tunable vacuum ultraviolet light, and they measured the threshold for ionization of these species, which was lower than of the stable isomeric forms. And so I guess as a nice story, I guess we could probably all share this. Reading this paper for the first time was highly motivating to me, and I immediately said, oh, we could do this in our laboratory, and I think there are other ways of detecting these systems besides photoionization, and so we started generating them in our lab, and so far we've made 4 different systems: the simplest 1-methyl, 2-methyl substituent, and an ethyl substituent, and there's by this same approach. I do want to emphasize that the chemistry changes a lot with substituent, and I'll touch on that, and we can come back to that perhaps in the questions or in future challenges. So what I realized and I wanted to investigate was that this carbonyl oxide group actually has an extended pi system, and I wanted to look for a very strong, what was predicted to be a very, very strong pi to pi star transition in these molecules. So I'm illustrating this here. There were theoretical calculations that I'm showing the molecular orbitals for indicated that there were 4 electrons in a pi system that's localized in this carbonyl oxide group. And there was predicted to be a very, very strong pi to pi star transition from bonding to antibonding excitation that would be similar to what's known in ozone, and which is isoelectronic. And so we went after trying to identify this very strong UV transition. So I'm not going to go into great detail about this, but basically what we did was simultaneous ionization monitoring. This is for the CH200, or ionizing using fixed frequency vacuum ultraviolet light in our laboratory. But then what we did is we introduced an ultraviolet laser, tunable, that would promote molecules from their ground state to an excited electronic state. And each time that UV transition was resonant, it would remove molecules from the ground state and therefore cause a depletion in our ionization signal. So you can see that on the right here. Here's the ionization signal. Here's the depleted ionization when we have UV light on, and here's the difference. And by using this technique, we could map out at different UV wavelengths the depletion spectrum, which is exactly equivalent to the absorption spectrum of these Criegee intermediates. And this is really important because this approach is now used for making measurements of the bimolecular reactions of these reactive intermediates with all sorts of other species, because there's a sensitive probe. And we extended that probe throughout all the systems, and we found that the absorption spectrum shifted a little bit, about 20 nanometers to shorter wavelength when we did alkyl substitution, and I'd be happy to discuss that. So let me get back to my main story for today. We really want to understand how alkene ozonolysis releases hydroxyl radicals, and this is sort of the origin of the title for my talk. I'm interested in understanding the reaction profile, the minima, the maximum, and how molecules move along this reaction coordinate to release hydroxyl radicals. So the figure that I have shown here is for the entire alkene ozonolysis reaction. There's a small barrier to get it going, but then it basically releases an enormous amount of energy as it goes through this first primary ozonide and then releases the Criegee intermediate. So it's so much better if we want to understand the mechanism if we can start directly from the reactive intermediate and follow the pathway out. And in particular, we're very interested in trying to determine the barrier associated with the rearrangement, the intramolecular hydrogen transfer reaction that ultimately releases hydroxyl radicals. So the way the mechanism works is if you have a methyl or an alkyl substituent, a hydrogen will transfer, an alpha hydrogen will transfer from, in this case, the methyl group to the terminal oxygen. That costs energy. We take energy to get up over a barrier to cause that hydrogen transfer to occur. And that, therefore, creates a new species, this vinyl hydroperoxide, after the hydrogen transfers. It has a lot of excess energy. The weak OO bond breaks, and it releases hydroxyl radicals. So for the rest of my talk, I'll just explain how we have been investigating this reaction coordinate. What's the barrier? What's the rate of reaction? And how does all of that change with substituents? So this is the way the experiment works in my lab. There were already theoretical calculations that identified the barrier associated with the reaction I just described. So let's take a look at that transition state. This is the transition state for hydrogen transfer from the methyl group to the terminal oxygen. You can see the hydrogen being shared between the methyl and terminal oxygen. And what we wanted to do was to provide enough energy to the system to drive this chemical reaction. So we initially started thinking that, this is all theoretical here, we were initially thinking that it would require 2 quanta of CH stretch in order to put in enough energy to surmount, or as I will show you, tunnel through that barrier. For the general audience here, tunneling, I guess I should just emphasize, tunneling means you're not going over the mountain, you're going through the mountain, okay? And it only works for light particles like electrons or protons or hydrogen atoms, which is the case here. Okay, so our experiments then are going to involve vibrationally activating this molecule. 1 quantum of CH stretch is not going to provide enough energy, enough energy to surmount. 2 quanta should take us right in the right region. And what we want to do is scan our infrared laser. This is a tunable optical parametric oscillator that, and we're going to then, basically as we tune, drive this reaction, and we're going to detect the hydroxyl radicals that are formed. Okay, so this was very, very exciting result. I don't know, it's the spectrum. Maybe it won't be so exciting to you, but really exciting result. What we were able to show was that by putting in, there are 4 different types of CH stretches in this molecule. They're color-coded here, and they're color-coded in the calculated spectrum. And what we would do is put in 2 quanta in any one of those modes, or 1 quantum in each of 2 modes. Those are called combination bands. And we expected to see 10 spectroscopic features in this region. And when we scanned our infrared laser and detected OH products, you can see that we generated a spectrum or a fingerprint of telling us that we were exciting this molecule, and it was recording out its infrared spectrum. But this is more exciting than just, putting your, just an FTIR spectrum. It's because each one of these vibrations is driving reaction, and we're detecting products. We're detecting the OH products that result. The part that was particularly surprising is that we were seeing features at significantly lower energy than we expected based on the theoretical calculations. We expected the barrier to be about here, and I'll show you in a moment that that's where actually it is. And we're seeing features to considerably lower energy, and even, I'll show you in a moment, we can go to much lower energies and the spectra, and still obtain spectra. So what's going on? Well, there's 2 possibilities. Look, either theory cannot calculate barriers very well. That's one possibility. You didn't hear me say that, okay? Okay. Or maybe, the result is that, or tunneling is occurring, quantum mechanical tunneling, and we're going through the barrier. So these are calculations. So this is our experimental spectrum for the methyl and the dimethyl substituted Criegee. In both cases, 2 quanta of CH stretch being put in. And the dashed lines indicate the best theoretical calculations to date of the barrier. And this takes into account zero-point energy. It's very fancy calculations, better than I can do, I assure you. Really excellent calculations. And so what you can see is that many of the spectroscopic features in this region, the infrared transitions, are at lower energy than the barrier, indicating that we must be tunneling. Okay, so we went from there and we said, okay, that's great. Maybe we can measure the rate of this process. What is the rate at which we're forming hydroxyl radicals? And we've done it so far for 3 systems, the methyl, dimethyl, and ethyl substituted Criegee intermediates in this same spectral region. So this is what the data looks like. Again, so color-coded so you can follow. We're measuring the appearance of OH products. It's happening on a nanosecond time scale. And it's happening with different rates for different substituents, okay? And we're exciting at a very similar energy. So there's a couple of reasons this could happen. They have different barriers, so that could affect the rate. The other things that are going on is that there are, the substituents are changing the properties of the molecule. So for example, you add an extra methyl group, you now have a low-frequency methyl rotor. So you're changing the density of states, and that affects the rate of dissociation. You add, in the ethyl group, similarly, you add an alkyl side chain, so you have low-frequency torsional motions, and again, this can have an influence on the rate of reaction. Okay, so I'm just going to give you a snapshot of the way we're thinking about things right now and tell you why I think this is really cool. So we can excite at different energies. They correspond to different infrared transitions of this molecule. And this is our experimental data for the methyl substituted Criegee. And what you can see is that the rate increases or the lifetime decreases as we go up in energy. And at about a year ago, we are collaborating with Stephen Klippenstein at Argonne National Laboratory, and he and simultaneously his group and our group were calculating from first principles the rate of this reaction. And the line I've just superimposed on our experimental data is the first principles theoretical calculation of this rate. There are no adjustable parameters. This is not a fit to the data. This is pure theory. And what it tells us is it tells us that we know very well what this barrier is and that we can predict the rate in a statistical fashion for this unimolecular rearrangement process. And I need to emphasize that the calculations are done assuming tunneling. So we got it right once. Let's do it again. This is what the data looks like in the next system. Again, theory is precisely, we can precisely calculate the rate of this reaction based on the barrier, and this is validating the barrier height. So what's the influence of tunneling? And this is part of the story that I want to emphasize. Theory can turn on or off tunneling, and so it's useful to think about or look at how different the rate would be, the rate of reaction would be in the absence of this of tunneling. And you can see that it's considerably slower and doesn't even occur at many of the energies we investigate. And so I just emphasize that the tunneling is coming about from this hydrogen transfer process, which is going through the barrier rather than over the barrier. So most recently, we've gone a lot lower in energy. And just to illustrate how important tunneling is in this system, and it'll take us into the thermal rates that I want to discuss next. So we went about 2,000 wave numbers below the barrier. Now we're exciting a CH stretch in combination with some other lower frequency mode. And again, you can scan the excitation, the IR laser, and be detecting products. And you can see that we observe several different IR transitions associated with this molecule. And then we go and look at the rate. And when we were up higher in energy, that rate was about a few nanoseconds. And now it's hundreds of nanoseconds. It's 100 times slower because it's that much harder to tunnel through the barrier at much lower energies. OK, so as an atmosphere, so we're now taking all the things we've learned from physical chemistry and now going to apply it to atmospheric chemistry. Atmospheric chemistry cares about the thermal rate. And so we wanted to then model, based on what we've learned from the energy-specific excitation and driving of this unimolecular decay process, we now have a very good determination of the barrier. We know tunneling is very important. And we're going to consider what happens in the atmosphere when you have collisions at 10 to the 10th per second, which corresponds to every 10th of a nanosecond you have a collision. So we can now model the thermal process just basically by having a Boltzmann-weighted distribution of the kinds of rates that I've shown you. And we can predict a thermal rate which is given here. But the key part is it's much, much slower, 6 orders of magnitude slower than what we were measuring, than what we've measured at higher energy. And it's so slow that it's actually very hard to measure because bimolecular reactions are happening simultaneously, and it's very, very hard to get a clean measurement of this. But nevertheless, and there's another possible channel. I should indicate that there's another intermediate along the path, and it's possible there could be trapping there as well. We don't know yet. So there have now been, since our experiments and theoretical predictions, there's now been 2 experimental measurements of this thermal rate for the dimethyl substituted Criegee, and you can see it agrees quite well. Well, when you can combine experiment and theory, you can not just measure it at one temperature, you can basically look at the thermal rate over all the temperatures that are relevant in the atmosphere and beyond. And so here this is all modeling based on our experimental data. And I want to emphasize this is just something that we've all learned in freshman chemistry. This is an Arrhenius plot. The log of the rate constant is a function of 1 over temperature, okay? And these are our predicted rates as a function of temperature. And you can see that we can predict, of course, the rate at room temperature for these different systems. It varies a little bit because the barrier heights are a little different. Calculated barrier heights are a little bit different. But the part that I want to emphasize is it's supposed to be a straight line, right? That's what we teach everybody. And the slope of that line is the activation energy. Well, it's not. It's curved. And that curvature is a signature of tunneling, even under the thermal rate. OK. So how important is this unimolecular process, the releasing hydroxyl radicals? It's an important source of hydroxyl radicals in the atmosphere, but it's in competition with bimolecular processes in the atmosphere. And the most important bimolecular process is with water vapor, okay? And experiments and theory done in other groups, including with Joe Francisco, who's here with us today, have indicated that for the simplest Criegee intermediate, this cannot undergo the mechanism I've been describing that occurs for the methyl substituted ones. The most important reaction in bimolecular reaction happens to be with water dimer. I think this is really quite surprising. At 70% relative humidity, this is it's 10 times faster than the rates that I was describing. And it was published by 2 groups in the last year. Joe Francisco and colleagues have just shown that, and this was just appearing, that the simplest Criegee intermediate actually also reacts on water droplets, so clouds, and even faster by 2 different mechanisms, a simultaneously hydrogen transfer reaction with the monomer, dimer, or trimer of water, but also in a completely different mechanism that involves proton transfer. For the methyl and dimethyl substituted and alkyl substituted systems, it seems that the loss is primarily due to this intramolecular hydrogen transfer process that releases hydroxyl radicals, and it's faster than bimolecular reactions in these cases. Okay, so I'm close to wrapping up. This is the picture of the atmospheric chemistry that I've come back to. What I've shown you today is that we've learned how to generate these reactive intermediates in the laboratory. We can measure the rates of unimolecular decay, and we can predict the thermal rates, and we can compare them with bimolecular processes with other species in the atmosphere. But before I leave you, I want to show you, I want to bring it all the way back to the atmosphere. And this is, atmospheric chemists, and this is done by Dylan Millay, who's at the University of Minnesota, can run global models to try to understand the chemistry. And what we're plotting here is the relative amount of hydroxyl radicals coming from the Criegee mechanism that I've been describing relative to the conventional mechanism, if you will, the solar-driven photolysis that leads to O singlet D plus water vapor. When it's red, that means that 40% of the reaction is coming from these Criegee intermediates. And so that's this reaction does not require sunlight. So of course in the winter time, the northern hemisphere is dark more of the time. And so the Criegee mechanism that I've been describing is quite important. You can see that it's also very important in regions with tropical forest regions where there's high alkene emissions. And of course, everything flips when it's the summertime. And although the tropical forests and biogenic alkene emissions are very, very important in generating hydroxyl radicals. Okay, so this is a brief summary of what I've said, but I also want to, I think I would want to focus for a moment on the future challenges. I think it's important to think about what next and what is missing. We've made a lot of progress, but we certainly don't know all the answers. And one of the things that's really important is that biogenic systems, the alpha pinene, the terpenes, we know nothing about their reactive intermediates. They have much more complicated side chains. Their chemistry is going to be different. And then another really important area for investigation is the initial stages of nucleation and growth of aerosol products in this chemistry. So I think these are important challenges to be thinking about. And I guess I just want to say that I want to thank the Dreyfus Foundation and many other groups and other agencies that support our work, and the team of scientists in my group that have done it all. So thank you very much.
We have time for questions.
Oh, hi. So in your comparison with RRKM theory with tunneling, the agreement is pretty remarkable, but still I see that the theory tends to underestimate the experiment a little bit. Do you know where that comes from?
Yeah, that's an excellent question. So I didn't go into the details about this, but so far we've been modeling the tunneling as a one-dimensional problem. And in fact, we've been using two different models. One is the simplest possible one. It's called an Eckhart. It's like an inverted parabola. And the other is, we also have been using Bill Miller's semi-classical transition state theory. But all of those methods are one-dimensional. And so one possibility is that there's coupling between the reaction coordinate and other degrees of freedom. The other issue is that the density of states is just very, very low. And so the number of states is rather sparse. And we think that also may be contributing. But all of these are important theoretical questions that we haven't yet explored. So, but it's certainly on our mind. And we are seeing deviations.
Jim?
So that was a very nice presentation. There's this abiding problem that observed OH in the troposphere is considerably higher than calculated.
Yeah.
So you've contributed slightly to the solution of that on this diagram. What's your judgment about the fundamental quality of the OH measurements?
In the atmosphere?
Yes.
Well, you know better than I, but I think there is controversy about the measurements themselves. I think some of the things that I'm following up on, the chemistry involving in pristine environments. This is making big news. I'm sure you're aware of it. In pristine forested areas, possible new sources or basically more OH out than went into the chemistry are really important questions. And I think that what do I think? I think there's potential problems in measurement, but I think there are actually big unknowns in the underlying science. So what do you think here? You need to comment briefly.
We don't have time to go over what I think.
That's one way of getting off of it. No, it's a great question. Okay, so of course, if you want to, I've tried to convince you that this is a hydrogen transfer reaction. Of course, you'll slow it down if you deuterate. Let me at least explain why it's a hard experiment that we're working on still, okay? So the way we've been injecting energy into the system is through CH stretches. So as soon as we go to CD stretches, the vibrational frequency drops a lot. So 2 quanta of CD stretch takes us at about the same low energy as I had been showing in one of my slides. And so we have the problem of getting the energy into the system because CD stretches are so much lower in frequency. And we predict, but we haven't yet measured, that it to be, I think it's like 50 times slower still at a comparable energy. We've deuterated, we've made the precursors. Actually, for those of you who know me, we're actually synthesizing molecules, okay? So we've created the deuterated systems, and we're trying.
The problem is not having like ether.
Okay, so that's another great question. It turns out that you basically you get local oscillators. They're so different in frequency that we can predict theoretically that they don't really communicate with one another. They're such different in frequency. But that's a possibility. Yeah, mixed isotopes. Yeah.
Let's thank Marsha again for a really interesting talk.